the last topic of the cytoskeleton, the microfilaments. बहुत ज़्यादा डिटेल में इसकी मैं नहीं जाऊँगा फॉर द रीज़न कि आपने इसको फिजियोलॉजी में पढ़ा होगा विद रेफरेंस टू मसल कॉन्ट्रैक्शन। तो हम इसको एज अ पार्ट ऑफ साइटोस्केलेटन देखेंगे और हमारा जो थोड़ा सा फोकस होगा वो इसके जो है रोल इन सेलुलर ट्रांसपोर्टेशन होगा। Microfilament, as the name indicates, these are at micro level as compared to the other two structures, intermediate and the microtubules. And their size is approximately 8 nanometers. They are responsible for multiple functions within the cell, like intracellular motility, phagocytosis, cytokinesis, and then within plant cell, they are the major cytoskeletal elements which are involved in cellular transportation as compared to microtubule, which plays role in the animal cells. So motility is more associated with माइक्रोफिलामेंट। इसकी रीज़न क्या है वो भी अभी हम इसको भी एलेबोरेट कर लेते हैं। If we look at its component of microfilaments, then it has two components, actin and myosin. पहले हम actin filaments की बात कर लेते हैं, because myosin जो हैं they are motor proteins just like kinesin and dynein, जो कि कार्गो ट्रांसपोर्टेशन का काम करती हैं, जबकि जो एक्टिन है दिस इज मोर और लेस लाइक नैरो रोड, नैरो पाथ, जस्ट लाइक माइक्रोट्यूब्यूल, जिसके अक्रॉस जो है वो मोटर प्रोटीन्स ने वॉक अराउंड करना है. So these are actin proteins which are present within the cell, and if you look at their structure, actin monomers, they polymerize, and for their polymerization we need ATP. Energy comes from ATP. So we have actin plus comes up the ATP, and then it gets attached to the actin filament on one side, or on the plus end. The polarization occurs here. The plus end is the fast growing end as compared to the minus end. Just like microtubule, there also exists the polarity of plus and minus end. Minus end is also called as the end where nucleation will occur. And as you remember, nucleation is the start of the polymerization, which is relatively slow process, and soon nucleation occurs. After that extension or polymerization, it is a quite speedy process. And the fast growing end is called as plus end, and the other end where nucleation has occurred is called as minus end. F-actin, actin filament, or microfilament. These are three different names or synonyms of the same structure. So we can see here that two filaments, they are making up a helical structure. और इस हेलिकल स्ट्रक्चर में अगर आप देखें तो इस सेंटर में एक जो है वो फरो है या कैविटी है which runs along the longitudinal axis of this actin filament. Then we can look at their stiffness level, that depending upon the cell type, these actin filaments, they can be loosely defined, they can be tightly anchored bundles, or they can have they are like have we can say have high order of arrays, like if for example if we talk about these epithelial cell जिनकी columnar shape है, so these filaments, they must be somehow tightly anchored so that the longitudinal shape of the cell can be maintained. On the other hand, if we have some circular cells and these circular cells have to have to pass through the narrow spaces, then these microfilaments, they will be loose so that the cell can change its shape according to the like artery or vein or capillary the cell is moving. So both the ends are present, whether these are loosely arranged, loosely or ill-defined network, or they are tightly anchored proteins. When we talk about its assembly, the for assembly we need an ATP, just like for tubulin polymerization we had GTP. So soon this ATP attached actin molecule comes and binds at plus end. It is converted into ADP ADP एक्टिन मॉलिक्यूल बिकॉज यहाँ पे डिफॉस्फोरिलेशन इसकी जो है हो जाएगी रेड कलर के जो प्रोटीन मॉलिक्यूल्स हैं we can simply isolate or identify them with reference to these orange colored one that these are attached having attached ATP and this yellow one is having attached ADP with these molecules. And as I earlier mentioned, the nucleation and elongation process. Then plus end को हम जो है एक और नाम भी देते हैं. This is called as barbed end, whereas the minus end is called as pointed end. This is just like an arrow. If you look at this structure, this straight line and the arrowhead and the tail, you can see here that this is an arrowhead, so a barb like structure, and the tail, this will appear like this. So barbed end as well as the pointed tail is the another example of polarity. Then comes the myosin. Myosin are of different types.
सर जो पॉजिटिव और नेगेटिव चार्ट है वो किस बेस पे था उनका?
ये जो है इसकी पॉलीमराइजेशन के हवाले से है कि जिस एंड के ऊपर पॉलीमराइजेशन हो रही है, एक्सटेंशन हो रही है, उसको हम ग्रोइंग एंड कहते हैं या प्लस एंड कहते हैं, और जिस एंड पे पहले न्यूक्लिएशन हुई थी जहाँ से फाउंडेशन बनी थी, दैट इज रेफर्ड एज नेगेटिव एंड. जी. Then comes up the myosin. There are different types of myosin proteins within the cell. All of these myosin except myosin 6 move towards plus end of the actin protein. Then these were first isolated in skeletal muscles as a component of actin myosin muscle contraction machinery, but later on it was found in different other types of cells. And if we look at the myosin, it is a motor protein just like kinesin and dynein protein, having a head region and a stalk and then a tail region. These head, stalk, and tail, they are made up of heavy chain of the proteins, whereas the neck region is covered by some light chain of the proteins. So this head region is basically the region which interact with these actin filaments, and this head region is having an ATPase activity because it has a site where ATP molecule binds, and then hydrolysis of ATP results in the energy generation. And that energy is responsible for movement of these myosin along with the actin protein. If we define or categorize the myosin, then we can group them into two structures: conventional and unconventional myosins. Unconventional myosins are further subdivided into different types based on amino acid composition. There are at least 17 different classes of unconventional myosin, type 1 and type 3 up to 18. Type 2 is not included here because type 2 has to perform a different function that is involved in muscle contraction. Now keep in mind, जब भी आप मायोसिन का जिक्र करेंगे, जब आप मसल कॉन्ट्रैक्शन में मायोसिन का जिक्र करते हैं, दिस इज मायोसिन 2. और इसके अलावा अगर हम बात करें मायोसिन 2 के अलावा जो है, वी हैव नंबर ऑफ अदर myosins present which are categorized as unconventional myosin, whereas myosin 2 is categorized as conventional myosin because of its discovery and the extent of its structure and function we have known so far. Then, keeping in view this amino acid sequences and structure, myosin 10 is found in vertebrates. Myosin 8 and 11, they are present in plants. When we talk about human beings, there are 40 different myosins that belongs to 12 different classes. So we can simply say कि हमने जो 17 different classes की बात की है, this is not mandatory that all these 17 classes are present in a single type of cell. There are different cells which have one type of myosin which may not be present in any other organism's cells. As the example shows, कि human they have 12 classes of myosin and 40 different types of myosins. When we come to the structure of this conventional or type 2 myosin, again we talk about its structure in detail, that there are 16 different myosin heavy chains, three of which function in non-muscle cells. That means 13 जो हैं they are related with the muscle contraction, and rest of the three they are not related with the muscle contraction, they are working somewhere else. Each myosin has a six polypeptide chain working or making the myosin as a motor protein. One pair of heavy chain, जैसे मैंने अभी थोड़ी देर पहले बताया, and two pairs of light chain. Heavy chain has one head portion, then tail portion, then stalk portion, and light chains they are like attached with the neck region. These heavy chains they are found in such a way that two myosins they are no not two, yes, two myosins, no sorry, this one myosin structure, and then the myosins are arranged in such a way, अगर इस structure की बात की जाए, this is a polar structure, head on one end and tail is on the other end, but these myosins they are going to work in the form of groups in such a way that their tails are going to interact with each other, as you can see here, and both the ends of these myosins they are having the head region. Therefore we call this kind of filament arrangement as bipolar filaments, and their heads can perform their functions independently or in like all together with one and other. And if we look at this particular picture, you can see this is one example of myosin 5. Myosin 5 is basically a motor protein that is involved in transport of vesicles within the cell. You can see here that this is the vesicle membrane, and this is the tail of the myosin 5, which is attached with certain protein on these vesicle, whereas the head region of this myosin 5 molecule is walking around along with the this actin filament. So the function of this myosin filament is similar to those of which we studied while studying the microtubule. That microtubule में हमारे पास जो kinesin protein है, that kinesin is responsible to walk along the microtubule. So microtubule का diameter जो है that is 25 nanometer. That means that this microtubule cannot extend up till the surface of the cell, or can go up till the plasma membrane because of its diameter. So in order to facilitate the transport to the terminal, to the membrane, plasma membrane, from this particular end the cargo is handed over to the myosin motor protein, which walks along with the actin filaments. And actin filaments, as they have very thin diameter, they are not as thick as the microtubules, so they have the ability to go up till the plasma membrane, and in this way the cargo transport is completed with the help of myosin protein and actin filament, along with the microtubules and kinesin. So we can simply say that transport is not only the function of microtubules and kinesin, but myosin and actin also plays its role for cellular transport. So this is all about your cytoskeletal elements and about your course contents we have completed today. Any question you can ask. Thank you very much.
ये दूसरी फिगर एक दफा
जी, दूसरी फिगर
एक दफा समझा दो।
कौन सी वाली, ये बॉटम पे है?
जी ये वाली दूसरी।
ओके, सो इसमें लेट मी टेक द पेन। जस्ट इमेजिन दिस इज द स्ट्रक्चर जिसकी मैंने बाइपोलर जो है मायोसिन की बात की है, एंड दिस इज
ये नहीं,
ये
ये जो ये वाला, ये वाला।
ये वाला। हाँ जी, तो इधर मैं आ रहा हूँ ना, पहले इसको मैं हाईलाइट कर लूँ। सो इस जो है बाइपोलर स्ट्रक्चर को सेंटर में से आप फोल्ड कर लें। when you fold it from the center, you will see this is going to be the shape. Head is interacting with the actin. Sorry, the yes, the head portion is interacting with the actin filament, और जो tail portion है that is free to interact with the cargo, or with the that is free to hold anything that has to be transported. If you remember, kinesin में भी हमने बात की थी, जो light chain वाला end है that holds or that lifts up the carrier, the cargo, that lifts up the cargo. So this is the situation when both the heads are interacting the actin filament, and this is one actin head is taking a step ahead from here. It has taken a step to the next level, and now this is one step ahead. अब नेक्स्ट स्टेप में क्या होगा कि ये वाला जो है हेड उठेगा और ये इसी तरह एक स्टेप आगे लेगा, एंड इन दिस वे थिंग्स विल कीप ऑफ, this protein will keep on moving along the actin filament. Now during this movement, if this protein is carrying a cargo on its attached on its tail, that is shown over here. आपको इस डिटेल में जाने की ज़रूरत नहीं है कि टेल किस किस प्रोटीन के थ्रू जो है वो वेसिकल के साथ अटैच है. आई एम नॉट गोइंग इनटू सो मच डिटेल. द ओनली थिंग इज दैट दिस इज द टेल पोर्शन which is going to get attached with the vesicle or cargo so that the transportation can be completed. Is clear?
यस सर।
Okay जी.